All right, and we're back with talking about macromolecules and our lipids. So the primary characteristic of lipids, which another way to talk about lipids is in terms of they are fats. So lipids are also fats. So this is why you hear of liposuction. So liposuction is for people that are morbidly obese and who have had problems with weight loss, that they can actually have the fat physically removed from their body. Liposuction is fat removal. So that's a way for you to remember which ones are lipids and which ones are carbohydrates, or what a lipid actually is lipids are fats. Lipids, the primary characteristic of a lipid is that it is going to be highly non-polar. So let's take a look at this hydrocarbon chain over here. We've talked about hydrocarbon chains in this class fairly frequently. So we've got a hydrocarbon chain, big old long structure made of primarily carbon and hydrogens. Well, as we know, we talked about this when we were talking about methyl groups. Methyl groups are highly nonpolar. Well, if you have a molecule that is made of not just a methyl group here at the end, but a huge structure of carbons and hydrogens repeating themselves, this is going to be extremely nonpolar. Now you might be saying, okay, well, yeah, it's nonpolar maybe, but Ms. Baer, there is a polar molecule here at the end. There is a carboxyl group here at the end. And carboxyl groups, as we learned when we were talking about functional groups, carboxyl groups are considered acidic. And that's true. The end of this molecule is going to be considered acidic. But when you have a structure that only has a couple atoms of acidity or a small region of acidity on the end of the larger hydrocarbon chain, the primary characteristic is not going to be the acidity, even though that's going to be important and it's going to come into play. The primary characteristic is that it's going to be highly non-polar. This is coconut oil. This is what coconut oil, the fatty acid typically found in coconut oil, lauric acid. So what we know about electronegativity and what we know about reactions at this point, this part of the molecule, this larger structure, this large non-polar structure here on the right-hand side, this is not going to react very well with water. It's not going to react to chemical reactions, right? It's not, if I put water into a solution with lauric acid, water and the hydrogens here are not going to be any more reactive to these carbons than they would be to they're going to want to stay with the water molecules, right? It's that like dissolves like characteristic, the idea that polar molecules dissolve other polar molecules and non-polar molecules dissolve other non-polar molecules. However, the end of the molecule over here does have a little bit of electronegativity imbalances. There's going to be some reactive possibility in the end of this structure. So keep that in mind. That's going to be really important. We're going to talk about this, and you've hopefully seen this in your videos and recognize it from your videos. Lipids, excuse me, lipids are primarily made of carbon, hydrogen, and then very little oxygen. There's definitely going to be some oxygen, as we were looking at with lauric acid, but the primary structure is going to be huge hydrocarbon chains. Lots of carbon and hydrogen. That's going to be the primary thing that's going to indicate to you that this is a lipid, is hydrogen and carbon. There will be some oxygen, but for example, if you compare this to a carbohydrate, carbohydrates have significantly more, there we go. Carbohydrates have significantly more oxygen on these structures. That's going to make them much more reactive, and it's going to differentiate them from lipids. Now, lipids are typically made, now we're talking about a specific type of lipid. We're going to go into the different types of lipids in a second, but typical lipids are going to be made of two monomers. You have the fatty acids, which you guys remember from your readings and from the videos you've watched. Fatty acids are going to come in two different types. You have saturated hydrocarbons, in which all of the carbons have as many hydrogens as they can bond with. And then you have unsaturated. You have unsaturated fatty acids in which there is some or multiple carbons that do not have all of the hydrogens that could potentially bond with them, right? They are not considered hydrogen saturated. And these molecules typically have a kink in them, which just means like a bend in the longer hydrocarbon structure. You're also going to have this molecule glycerol. This glycerol is technically not a lipid. Not a lipid, right? This glycerol molecule is technically a carbohydrate because look at all of this oxygen. Look at all of these hydroxide groups here, right? These hydroxide groups are going to make it very reactive. Hydroxide groups are going to make it very reactive, and it's no longer considered, or never was considered, a fat or a lipid. It's considered a carbohydrate. But as we said when we were talking about carbohydrates are typically the building blocks of larger molecules. So the molecule that we just described was a triglyceride fat. So when we go back to this structure, the two structures that we're concerned with are triglyceride fats. This is going to be the primary, when you think of fat in foods, when you think of butter, when you think of coconut oil, when you think of olive oil, these are your triglyceride fats, right? Triglyceride fats are oils or butters, right? Things that are primarily found in food. The interesting thing is that the glycerol molecule we were talking about a second ago, the glycerol molecule we were talking about is right here. This is that original carbohydrate structure. And this molecule had to undergo a chemical reaction. This molecule had to undergo a chemical reaction in order to attach glycerol and all of these fatty acid chains, right? These are fatty acids. And there are 3 fatty acids attached to glycerol. This is important to notice. How did these molecules get attached to each other? How did these molecules end up attached to each other? It is because of the acidic carboxylic acid group at the end. The carboxylic acid group at the end is undergoing a synthesis reaction with glycerol, right? So if we go back to that slide that we had just a second ago, glycerol and fatty acids are going to undergo a synthesis reaction to create triglyceride fats. So you're going to need 3 fatty acid chains and 1 glycerol in order to make fatty acids. Okay, in order to make fatty acids. This is going to be important for when you're doing your macromolecules assignment. The second type of lipid that we have is this thing called a phospholipid. We're going to talk about this more in class, but the phospholipid is this guy. You guys see the long non-polar tails. You've got one with a kink right here. You've got a saturated and an unsaturated fat. You've got what appears to be something like a glycerol molecule, right? Another glycerol molecule. But then phospholipids, this is where the phosphorus comes in. You've got a phosphate group here, and then you've got this polar head on this side. This is just a molecule that happens to have a nitrogen in it, and that creates polarity in this structure. So you've got this polar head. The big characteristics you guys need to pay attention to are the fact that you've got these huge non-polar, what we call tails, and a polar head associated with this. And this is going to be really important. This is going to be really important. We actually talked about a structure like this when we were talking about soap and the cells that soap makes around non-polar molecules. Phospholipids are going to do something very similar. Phospholipids are going to be the primary molecule found in cell membranes. Phospholipids are going to be the primary molecule found in cell membranes. This is just important to remember. We're going to talk about it more when we talk about cell membranes in the cell unit. Last type of lipid that we have, the last variation we have is the steroid structure. So if you remember, we talked about how each bend in the structure is going to represent a carbon. And because scientists are lazy and they don't want to draw all of these carbons, not necessarily lazy, I would call it efficient, right? You don't want to draw all of these carbons because even these carbons are going to have hydrogens attached to them as well. So it's simple, it's significantly simpler to just draw the bends in the structure. But what you can tell just by the carbons that I just drew, there is a ton of carbon and hydrogen in this structure. Ton of carbon and hydrogen in this structure. You've got a couple oxygen molecules, which means this molecule can create chemical reactions. It is going to undergo some sort of chemical reactions, but the larger ring structure that makes up the bulk of this molecule, is going to stay stable. It is not going to undergo chemical reactions. Steroids are hormones. Steroids are going to be things that flow through our body, and then they create chemical reactions to either manipulate our brain or manipulate other parts of our body. So it's going to be really important that they are able to undergo chemical reactions, but that they aren't completely digested by the body's digestive system. And so that largely stable chemical ring structure is going to be really important for maintaining that characteristic, right? We're going to go into all three of these things in a lot more detail in Unit 3. And that's it. That's what we've got for lipids. And keep an eye out for the next one, which will be nucleic acids.